Hey everyone, Dr. D here, and in this video we're covering Chapter 5, so let's get started. So Chapter 5, what we're looking at is the membranes of the cell. Specifically, we're going to be mainly looking at the plasma membrane of the cell. We're going to talk a little bit about the structure of the membrane and the function of the membrane. Now, you should know that the plasma membrane obviously is the boundary that separates the outside, the extracellular stuff from the inside, the intracellular stuff of a cell. And so what's inside of a cell is the cytoplasm and all the organelles, and what's outside of the cell is called extracellular environment. Now, the main function of the membrane, obviously besides keeping whatever's inside and whatever's outside is to be selectively permeable. What does that mean? It means to allow certain things in and certain things out. Not just anything should be able to come into the cell, and not just anything should be able to float out of the cell. The cell needs nutrients to come in. It also needs waste to come out. You know, all of this requires the membrane to be selective in its permeability. That means selective in what it allows in and what it allows to leave the cell. Okay, now remember that phospholipids are partly polar and partly non-polar. This means the term for that is amphipathic. Amphipathic means a molecule that has a polar and a non-polar end. Do you guys remember what phospholipids look like? They have glycerol, 2 fatty acid tails, and a phosphate group. So they kind of look like this. Remember, this is a review of a previous chapter, chapter 3. You've got a glycerol, you've got 2 fatty acid tails, and you've got a phosphate group. You also have this little variable group up here called R. Usually that's choline, but you don't need to know that. What you do need to know is that the head part of the phospholipid, and again, we've already learned this, the head part of the phospholipid is hydrophilic because of the charge on the phosphate group, and usually this variable part, this R group, has a charge as well. So the head part actually mixes with water, whereas the tails do not. The tails are hydrophobic. Okay, and because this molecule is part polar, or actually charged, I should say hydrophilic, and part hydrophobic, it's considered an amphipathic molecule. Okay, that's what amphipathic means. Now, remember what phospholipids do? They all kind of line up side by side, okay, in a bilayer. So you've got 2 layers of phospholipids, and that makes up a membrane, including the membrane of the cell, the plasma membrane. Okay, so for example, if you look here, this would be the outside of the cell, for example, and down here would be the inside of the cell, for example. So you've got the hydrophilic heads pointing to where the water is, either inside of the cell or outside of the cell. Now you see where the tails are? That's called the hydrophobic region. That's where water isn't. There shouldn't be any water in here with these tails, because these tails, these fatty acid tails, are just long chains of basically carbon and hydrogen, right? Hydrocarbons. There shouldn't be any water in here with these tails, okay? So membranes kind of look like this, right? And what you need to understand is that membranes are not frozen. You see, they're not static. What do I mean by that? Do you guys remember I said in a previous chapter that, I think it was chapter 3, we discussed that lipids do not attach covalently to other lipids, so you don't have this lipid stuck to this lipid. So if you look at the phospholipid bilayer here, none of these phospholipids are actually connected to one another. None of them are covalently attached. You know what that means? That means this phospholipid right here, you see the one I'm highlighting? This phospholipid right here is free to mosey on this way, and its neighbor is free to go ahead and hang out and go the other way. So none of these phospholipids are attached to one another, and none of them are glued in place. So this one can float away that way, the other one can float away the other way. So this is a fluid membrane. Fluid means there's movement involved, right? And the membrane is a fluid model. But not only is the membrane a fluid model because the phospholipids are free to float around, but it's a mosaic. And what do I mean by mosaic? Mosaics are composites made up of different parts, right? Like artwork made up of various shards of different glass. Well, here you don't have shards of glass. You have different proteins. You see these blue things in the membrane? Obviously, you have a membrane, a phospholipid bilayer. But a membrane is not just a phospholipid bilayer, especially your plasma membrane. It's not just a phospholipid bilayer. You also have all kinds of proteins that exist inside the membrane. You see, you also have other stuff in the membrane like this. This is a steroid called cholesterol. You see, you could have cholesterol in the membrane, which is a steroid. You can have proteins in the membrane. Some of these proteins span the entire membrane. We talked about these before. These membranes, these are called transmembrane proteins because they span the whole membrane. And then there are other proteins that are more loosely associated with the membrane. But anyway, you see how you have all these different components in the membrane? Well, that's called a mosaic, right? You've got a mosaic in the membrane. The membrane is actually a fluid because the phospholipids float around. Mosaic of all different components model. Okay, so the membrane is considered a fluid mosaic model. And all of these proteins, unless they're anchored to something, these proteins are free to float around laterally. Laterally means, left and right and in space, kind of in one dimension or two dimensions in space, left, right. These proteins are free to float around in like icebergs in the ocean, right? Because they're not stuck in place, unless, of course, like I said, they're anchored to something, which some proteins are anchored. Okay. Now you might be wondering, what is this character here, this little cholesterol? Remember I said there's steroids in your membranes called cholesterol? There's a reason you have steroids in your membranes, the cholesterol in your membrane. It serves as a fluidity buffer. What does that mean? Well, you know what fluidity means, right? Keeps things moving when things move around. And do you know what a buffer is? A buffer is something that keeps, it's a substance that keeps conditions set, right? So if you're talking about a fluidity buffer, what we're talking about is something that keeps the membrane fluid if it's cold and keeps the membrane fluid if it's hot, right? That's a fluidity buffer. So when it gets too cold, the membrane wants to freeze, but cholesterol keeps the membrane moving instead of freezing. If it gets too hot, the membrane wants to break apart from too much movement, and what it can do is suppress some of that movement, keeping it from breaking apart. And by the way, these cholesterol steroids I've been telling you about, they exist in the membrane of cells, but only in animal cells. Okay, you find them in animal cells, and animal cells have saturated fatty acid tails. You see, animal cells are much more prone to freezing when it gets cold than plant cells because of those saturated fatty acid tails. Remember one thing about the saturated fatty acid tail, one property is that causes freezing at relatively high temperatures. Well, the cholesterol molecules, these steroids, keep the membrane moving. It's a fluidity buffer. Now, plants, on the other hand, if you look here at the top left, plants have unsaturated fatty acids. And so their membranes are fluid, and they don't actually require any steroid. They don't require any cholesterol in the membrane. So you'll see that plant products do not have cholesterol, but animal products do have cholesterol, and that's one primary reason. Animal cells need that cholesterol in there to keep the cells fluid, whereas plant cells have more of a natural way with the tails of keeping the membranes fluid in cold conditions. Okay, that's why if you go into your pantry and you look up, you find vegetable oil, olive oil, peanut oil, coconut oil. You look at the back of all those bottles of the nutrition facts, and you'll see that there's no cholesterol in any of those plant products. Only animal products have cholesterol. And why do we have cholesterol? It serves as a fluidity buffer to keep our membranes fluid in cold and hot conditions. Okay, so moving on, you might be wondering, what are all these proteins in the membranes? Do you remember these proteins? And what are all these proteins doing in the membrane? Some of these proteins span the whole membrane. That's called the transmembrane protein. And some of those proteins don't span the whole membrane. Those would be called non-transmembrane proteins. But either way, they're membrane proteins. One other thing you should understand is integral membrane. What does integral mean? Integral means that the protein goes deep into the hydrophobic core. You know, this hydrophobic area of the membrane where the tails are hanging out. So transmembrane proteins that span the whole membrane, they're by definition integral proteins, whereas non-transmembrane proteins that don't span the whole membrane, those are usually peripheral membrane proteins. That means that they could be sometimes washed out, washed away. They're not so ingrained into the membrane of the cell. Okay, so what are the jobs of these transmembrane proteins? I've been saying transmembrane proteins, and you see what they do. They span the entire membrane, so they are integral membrane proteins. So what are the jobs of those transmembrane proteins? Well, there are several jobs for transmembrane proteins. Here's one. Transporters, right? Transmembrane proteins can transport things into or out of the cell through a hole or something, right? Here's another job, an enzyme. Transmembrane proteins, some of them can act as enzymes, changing a substrate into a product, catalyzing a reaction. Here's another job. In this case, the transmembrane protein is serving as a receptor. Here's a ligand hormone. This green structure would represent a hormone. The hormone attaches to the receptor. The receptor then signals into the cell so the cell can do something. You could have transmembrane proteins that are identity markers. They're here to identify the cell. So, for example, your white blood cells might want to know, is this a self cell or a foreign cell? Because it might want to destroy the foreign cells in your body. Well, the identity markers on the cell will help to distinguish that. Other transmembrane proteins could serve to adhere one cell. So here's the membrane of one cell to another cell. Here's another membrane on another cell. You see, so you could have one transmembrane protein from this cell fastened to a docking site, basically on another cell, transmembrane protein. This would be called cell-to-cell adhesion. It explains why you have, your tissues or cells that are really stuck to one another. That's because you actually have proteins between the cells like this, holding the cells together. And of course, you can have transmembrane proteins that specialize in attachment to, basically anchor proteins to attach either to the cytoskeleton or to the extracellular matrix. You guys remember in the last chapter we talked about, in chapter 4, we talked about integrin. Integrin was an example. Integrin was a transmembrane protein that attached to actin microfilaments inside of the cell, and it actually also attached to the extracellular matrix. Remember the collagen, the proteoglycan complex. So you can have all kinds of transmembrane proteins that are involved in attaching to things. Okay, so this is just 6 examples of typical jobs for transmembrane proteins. Okay, this is just showing you the complexity of these transmembrane proteins. Some of them can consist of one alpha helix. Some of them are more complex with multiple alpha helices. In fact, G proteins have 7 transmembrane alpha helices. You could make a basket out of beta sheets, right? So this is called a beta sheet basket. A lot of transporters are made out of this beta sheet pattern. So this, don't let this confuse you. This is a slide just showing you that these transmembrane proteins can span from, they're diverse. They can span from very simple to highly complex structures, and very small proteins to very large complex proteins. Okay. Now, what else? Here's the thing. Where did these transmembrane proteins come from? So if we're looking at the plasma membrane of the cell, you see what I'm highlighting here, the plasma membrane of the cell. Where did these transmembrane proteins come from? So remember where they all originated? They originated at the ER, the endoplasmic reticulum, specifically the rough ER, the rough endoplasmic reticulum, right? If you look closely here, transmembrane proteins are made into the membrane, directly made into the membrane of the rough ER by ribosomes at the rough ER. Those proteins then leave the rough ER through transport vesicles. Those transport vesicles fuse to and become with the cis face of the Golgi apparatus. And then those transmembrane proteins are modified as they make their way to this trans face. So they dock at the cis face of the Golgi apparatus, and then they make their way to the trans face of the Golgi apparatus. And along the way, look, the transmembrane protein, look at it. It's along the way it's getting modified, right? In this case, it has a little green sugar attached. You see this green structure? It looks like little antlers. These green structures are sugars that are attached to the transmembrane protein. So the transmembrane protein was modified, in this case, by addition of sugars. Then the Golgi apparatus at the trans face will start to pinch off into a secretory vesicle. That vesicle will transport it to the plasma membrane, where the vesicle fuses with the plasma membrane, delivering the modified transmembrane protein to the plasma membrane. That's how the transmembrane protein got there. And do you notice how it's called the transmembrane glycoprotein now? Well, you know what glycoprotein is? It means that there's a sugar attached to the protein, and that sugar was attached by the Golgi apparatus. But I mean, it's just a good general term to know. Whenever they're talking about glycoproteins, I mean a protein that was modified with a sugar, and usually the Golgi did that modification. A glycolipid, conversely, a glycolipid would be a lipid with a sugar attached. A glycoprotein is a sugar with a glycoprotein is a protein with a sugar attached. A glycolipid is a lipid with a sugar attached. It's just you don't need to understand it too much in depth right now. But later on, when you take biochemistry, it'll become more clear why you have to care about glycoproteins or glycolipids. Okay. So again, the membranes are selectively permeable. They allow certain things into the cell. They allow certain things out of the cell. Now, what you need to understand is there are some molecules that can cross the membrane regardless of transport proteins or not. These molecules will cross the membrane directly, almost as though the membrane doesn't exist, okay? And what you need to know is that those molecules that can just cross the membrane as though the membrane doesn't exist are nonpolar molecules, hydrophobic molecules, and usually small nonpolar molecules, okay? Examples are hydrocarbons, oxygen, CO2, right? So small hydrocarbons, I'm talking like CH4, right? No, methane, oxygen, CO2. These are the types of small molecules like N2, nitrogen gas, H2, hydrogen gas. Any kind of small nonpolar molecule has an easy time crossing the membrane. Now, what do I mean by crossing the membrane? Let me show you. You see this membrane here? I'm talking about stuff that can just cross the membrane as though the membrane doesn't exist. It doesn't need a transporter. You see, it doesn't need a transporter. Just molecules that can just cross right through the membrane, right through the phospholipids, right through that those tails. The hydrophobic domain where those tails exist. I'm talking molecules that can just cross the membrane as though it's not even there. The only molecules that can cross the membrane and just cross like nothing's there are small nonpolar molecules such as methane, oxygen, CO2, nitrogen gas, hydrogen gas, those types of things. Okay. Now, what if we're talking about molecules that aren't nonpolar? What if we're talking about polar molecules or charged molecules? Then what? Can those molecules cross the membrane? Let me show you another example real quick. Let me show you real quick so you understand this a little better. Let me go back to this. So for example, let's just look at this top right example. Remember, what could cross the membrane directly as though the membrane doesn't exist? Small molecules such as O2, CO2, methane. These are the things that can cross the membrane as though the membrane doesn't exist. All right, they just cross right across the membrane. Okay, but what if I have a polar molecule such as I don't know, glucose, right? Glucose. Let's say my laser dot right now is glucose and it wants to cross the membrane. What happens? It's polar, right? So it's floating around the sugar. I'm sorry, it's polar. It's floating around the water. So that glucose, that sugar tries to get in. What happens once it tries to get into the membrane? What are all these tails? These tails are nonpolar. Do polar and nonpolar things mix? No, so my sugar will get bounced off. You see, so my sugar tries to go across. My sugar gets bounced off. It can't make it through this giant area, this hydrophobic region, this space where, there's a bunch of carbons and hydrogens. Polar things and charged things can't cross. So, for example, you know the types of things that can't cross? Polar things like ammonia, I don't know, water. Water can't even cross. Look, water molecules can't even cross this membrane. You know why? Because they're polar. So let's say this laser dot is water. Water's going to try to cross. It's going to find all these tails. It's going to bounce right back out. You see that? Water doesn't cross this membrane because water's polar. Ammonia can't cross this membrane because ammonia is polar. Glucose can't cross this membrane because glucose is polar, right? So polar molecules can't cross. What about charged ions? What about ions? Remember, like sodium ion Na plus or chloride and Cl minus. Can those cross the membrane? Na plus. Let's say I'm Na plus. I'm trying to cross the membrane. Nope, my charge is not going to want to mix with these nonpolar tails. What if I'm Cl minus? Chloride, can I cross the membrane? No, I get bounced right out. So you see, only nonpolar small molecules can cross the membrane. But what if the cell wants glucose? What if the cell wants water to come in? What if the cell wants sugars to come in or sodium to come in? Then what? Well, in that case, you're going to need transport proteins. That's why we have transport proteins. You're not going to need a transport protein for small nonpolar things like oxygen or CO2 or methane because there is no transport proteins for those things. Those things just float across the membrane as though membrane doesn't exist, right? But what if you want to bring in sodium ion or glucose or water? Well, then you're going to need transport proteins. These proteins allow polar things and nonpolar things to cross the membrane, okay? Some of them are called channel proteins, and the channel protein that allows water to cross is called an aquaporin. Some of them are called carrier proteins, and these allow things to cross. So what's the difference? Let me show you the difference real quick. Okay, channel proteins are transporters, and carrier proteins are transporters. But what's the difference? Look, at the top you have a channel protein. Look at the solute. The solute's going to try to cross the membrane. And now let's say the solute, you see this orange dot here? Let's say this is glucose, right? Can glucose cross the membrane directly? These dots, can they cross the membrane directly if it's glucose? Can it cross right through the membrane? No, because glucose is polar, right? And polar molecules can't cross the membrane. But if I have a transporter, oh look, now glucose can cross, you see, because the inside of the hole is polar. You see what I'm saying? The transport protein has a polar area inside of this donut hole. So the molecule can actually cross through the little donut hole. That's called a channel. Basically, a channel. It's what it sounds like. It's just a little channel. It's like a little hole, a little pore that allows solute to cross. Do you guys remember what solute means? Solute is the thing dissolved in the solvent. That's important to understand. Solutes are the substances dissolved in the solvent. So, for example, if I have sugar water, the sugar is the solute, the water is the solvent. Solutes dissolve in solvents, okay? Now, if you want glucose to cross the membrane, that means that the solute is polar. You're going to need a transporter to allow that polar thing to cross the membrane. So what if the glucose wants to cross a different way? It doesn't have to be a channel. You could use what are called carriers. Okay, in this case, the glucose wants to cross. How does it cross? It has to actually interact with the carrier. It's not just a hole that it crosses through. Carriers actually interact with the solute. When the solute interacts with the carrier changes conformation. What that means is it changes shape, allowing the solute to cross. You see, so the only difference between channels and carriers is that they're both transporters. Channels are basically pores or holes that allow the solute through. Carriers actually change shape to allow the solute through. Okay, and if you really want to know, channels are usually for smaller solutes to cross, and carriers are usually for larger solutes to cross. Usually, that's like a rule of thumb thing, but it's not. It's not in every case. Okay, there's actually a channel that's specifically for water. By the way, there's a channel just for water. It's called the Aquaporin. Look at this slide here. Let me show you. Aquaporins. Aquaporins are basically a channel that facilitates the passage of water. Again, remember, water can't just cross the membrane directly. It needs help. So aquaporins are special channels that allow specifically water across. So another thing you need to understand is there are very specific channels for very specific substances to cross the cell. So for example, you've heard of potassium channels maybe? Well, potassium channels specifically allow potassium ion to cross. You've heard of chloride channels? Chloride channels specifically allow chloride to cross. Aquaporins specifically allow water to cross. So these transporters aren't just big old holes that let just anything cross the membrane. Remember, the membrane is selectively permeable. It doesn't just let anything across. These transport proteins are specific for their substances or their solutes that they allow across. Okay, so let me take a quick break with that. When we come back, I'll explain what drives a substance across the membrane. Like, do you need power? Do you need energy to move something across the membrane, or is it spontaneous? Do things cross the membrane spontaneously? We're going to talk about that when I come back. So let's take a quick little break. All right, back from break time. Let's go ahead and talk about passive transport. Well, let's first discuss diffusion. We need to understand how diffusion works in order to understand why things cross the membrane of a cell. So what is diffusion? Diffusion is the movement of molecules from high concentration to low concentration. So if I were to release a gas right here, that gas would be high concentration right here. Those molecules would move away. They'd move towards lower concentrations of gas, and that's diffusion. And it doesn't have to be gas. You could put, like in this picture here, look at your image here, a drop of ink. You put a drop of ink in a beaker, that drop is a high concentration of ink or dye. Those molecules are going to separate, and they're going to spread out toward lower concentrations of dye until all of the dye molecules are equally spaced apart. Now, what you need to understand is that diffusion is a spontaneous process. Do you guys remember what spontaneous means? So hopefully you do from chapter 6. It was on the last exam. But chapter 6, we covered spontaneous. That means negative a delta G. That means happens for free and releases energy, right? Happens spontaneously without the input of energy, and in fact it releases energy. And you do need to understand that diffusion releases energy. Diffusion is spontaneous. Diffusion happens. It just happens. It's spontaneous. It's driven by entropy. Okay, another thing you need to understand is that diffusion, when it happens, when we say something is diffusing, what we say is that the molecules are moving down their concentration gradient. So when I say those molecules are moving down their concentration gradient, what do I mean? I mean that the molecules are moving from high concentration toward low. And that's the way that diffusion works. The molecules move down their concentration gradient. Now, if you were to try to go against that, that's called going against the concentration gradient. So diffusion means molecules move down their concentration gradient for free. That's negative delta G. If you were to try to put those molecules all back together again and bring them all together again, well, that wouldn't be diffusion. That would be molecules going from low concentration, trying to force them back to high concentration. That would be going against the concentration gradient, and that would be a positive delta G, right? That wouldn't be free. Okay, going down the concentration gradient, that's diffusion. That's negative delta G, that's free. That releases energy. Going against the concentration gradient, that's not free. That's positive delta G. That is going to require energy to do. Okay, so now, all right, so I've spoken a lot, but what am I trying to get at? Now, molecules can either diffuse across a membrane, right? Molecules, we're talking about if there's a membrane, molecules want to cross the membrane. Now, the question is, are those molecules going to cross the membrane for free? Is it spontaneous? Is it driven by diffusion? Or are we trying to move things across the membrane against the concentration gradient? And is that going to cost us, energy? Is that going to be not free? So let's talk about free first. Let's talk about when molecules cross a membrane for free. This is called passive transport. Passive transport is movement of molecules through the membrane in which A, no energy is required, and B, molecules move in response to a concentration gradient. So let's say I have a membrane. If substances cross the membrane down their concentration gradient, that's just diffusion doing its job. That didn't cost me any energy. That was spontaneous. That was negative delta G. And in fact, that released energy, if anything. Okay, so let me show you some examples of this. Okay, in this example, these molecules of dye, these orange molecules of dye, they're really high concentration on the left side of this picture, aren't they? And do you see what this dashed line represents? This dashed line represents a membrane, okay, a membrane. And do you see there are pores in the membrane, holes in the membrane for the dye to cross? The dye will cross the membrane. Now, so basically diffusion can happen, right? These dye molecules can cross from where there's a high concentration of dye towards where there's a low concentration of dye, right? So you see the dye can go from the left side to the right side, from high concentration on the left toward low concentration on the right. So what do these dye molecules do? They cross from left to right, down their concentration gradient. And do you see the pores in the membrane? Those pores are big enough to allow the dye to cross. So which way are these solute molecules going to move? Which way are these dye molecules going to move? Well, you have to ask yourself, where's the high concentration of dye? Obviously, it's on the left. Where's the low concentration? In this case, no concentration of dye is on the right. So the molecules are going to diffuse from left to right. You see that? These molecules of dye are going to move from left to right. So basically what they call this is a net diffusion. You know what net diffusion means? That means more molecules are diffusing in this direction than the other direction. So that doesn't mean molecules can't diffuse back to the left. What net diffusion means is that there's many more molecules diffusing to the right than are diffusing to the left. That's what net diffusion means. Now, once you have the concentrations equaled out, okay, so there's an equal concentration of dye on the left and an equal concentration of dye on the right. At this point, you have reached what's known as equilibrium. In this case, it would be considered a delta G equals 0 situation. Remember equilibrium. What that means is there is no net flow of these dye molecules. For every one dye that moves to the right, one dye will move to the left. So there's no net flow. And because of that, you have reached equilibrium, which means delta G equals 0. However, before that, before you reached equilibrium, you were a negative delta G reaction going on here because the molecules were net diffusing from left to right down the concentration gradient. Okay, so hopefully you're following along with all of the terminology here. I know it's a lot of terminology, but once you get it, I assure you it'll become very, very straightforward. Okay, then look at the example at the bottom here. This is interesting. In this case, you have two different dyes. You have an orange dye on the left and you have a purple dye on the right. And you know what's really interesting? Even though you have a higher concentration of orange dye on the left than you do of purple dye on the right, what you need to understand is that something special happens here. The orange dye kind of pretends like the purple dye doesn't exist. They both diffuse down their own respective concentration gradients. What does that mean? That means that the purple dye net diffuses to the right because there's a higher concentration. I'm sorry, the orange dye. The orange dye has a higher concentration on the left, so it's going to diffuse to the right. And the purple dye ignores that. It just cares about itself. The purple dye, all it knows is that it has a higher concentration on the right, so it's going to net diffuse to the left. So the moral of the story here is that when you have different solutes, different types of molecules, they each diffuse down their own respective concentration gradients. The orange dye will diffuse to the right, and the purple dye will diffuse to the left. They ignore one another as though the other one doesn't exist. Okay, so that's kind of interesting and worth understanding there. Okay. And they're both going to eventually reach equilibrium. All right, what else? So, oh, by the way, let me tell you something here. This is an important term to know. You see what's crossing the membrane here? What's crossing the membrane in these examples here on this slide? Is the water crossing the membrane, or is the solute? Is the solvent crossing the membrane, or is the solute crossing the membrane? Think about that for a second. It looks like the dye molecules are crossing the membrane mainly, right? So isn't that the solute, right? The dye molecules are the things dissolved in the water, the solvent. So why do I point that out? Well, because when solute is crossing the membrane, all right, you guys, when solute is crossing the membrane, this is called dialysis, okay? You need to know that's called dialysis, okay? When that's happening down the concentration gradient, that diffusion is moving a solute across the membrane, that's considered dialysis, okay? That's a solute transport across the membrane. All right, so these examples on this slide would be dialysis problems, right? Dialysis, where the solute is crossing the membrane. Now, what about this? Look at this example here. This is very similar type of setup, but there's a critical difference. Let me show you. Here's a tube shaped like a U, and the blue stuff's water, obviously. The green dots are, look, it says sugar molecules. The green dots represent sugar. Now, you also have a membrane at the bottom here. You have a membrane. If we zoom in, you see the arrow here? We're zooming in on that membrane. What do you notice about the membrane? Take a quick look real quick. What do you think? It looks like there's a little arrow that looks like water. This is a water molecule. Water molecules can cross, you see? But do you see the green dots crossing the sugar molecules? Are the sugars crossing the membrane? No, because look, the pores in the membrane here, this membrane is selectively permeable. The pores in the membrane are too small, too fine to allow the sugar to cross. So you see what the problem is here? Look, in this example back here, the pores were plenty big enough to let not only water across, but the solute across, the dye molecules to cross, right? So the dye molecules crossed, right? Look over here. Can the sugar cross? No, the sugar can't cross this selectively permeable membrane. So now we have a different type of problem set up, don't we? Now we have a different type of problem. Now let's take a look. All right, let's try to figure out what's going to cross where. So if the sugar could cross, first of all, let's ask this. If the sugar could cross the membrane, which we know it can't, If the sugar could cross the membrane, which way do you think the sugar would go? From left to right? Or do you think the sugar would cross from right to left? What do you think? Think about that for a second. Which way would the sugar cross? Well, you have to ask yourself, okay, well, the sugar would cross through diffusion, and that would be passive transport. That would be happening for free. And remember what diffusion means? Substances move from high concentration to low, right? So where are the green dots high concentration? Do you think you have a higher concentration of green dots of sugar on the left or a higher concentration of sugar on the right? And do you guys remember what concentration means? I don't think I mentioned this, but concentration is, you can think of it as number of green dots per area, right? So if we look at this area here, look, what I'm circling on with my laser pointer, this area, you see there's only 3 green dots, whereas if I put a area the same size on the other side, you see there's 1, 2, 3, 4, 5, 6. There's 6 dots in the same area. So things like grams per liter, those types of things, or moles per liter, these types of things are concentration. Okay, concentration. Concentration means amount of solute per solvent, in this case. Concentration, amount of solute in solvent, in a set amount of solvent. So where do you have a higher concentration of green dots? Obviously on the right. And so which way would your green dots want to diffuse? The green dots would want to diffuse to the left. They would want to net diffuse to the left, correct? But can they do that? Look at your membrane here at the bottom. Can those sugars diffuse? They can't. They can't. So, do you see the problem here? The conundrum. How this is a different problem than this one we did earlier. In the first example, in this one, the solute could cross the membrane. So the solute would cross from where solute is high concentration, and there would be net diffusion to where solute is low concentration. Do you see in this example here, the solute cannot cross. It is not possible for the solute to cross the selectively permeable membrane. So what happens in this case? Nothing? Well, I can tell you something does happen, and that is, take a look. The water will cross. The water will cross. And do you see, it looks like 3 water molecules are crossing to the right, and only 1 water molecule is crossing to the left. So there's a net flow of water from left to right. Now you might be asking, wait. Why is water going from left to right? Isn't there a higher concentration on the right? This is a very, very, very common stumbling block for students. This is something that trips students up. So let's think about this real quick, okay? This is going to make sense of it, so pay attention to me right now. Look, do you see the green dots? What are the green dots? That's the solute, right? Those are the sugar molecules, which is the solute. If the solute is the one doing the diffusing, okay, do you guys follow me? If it's the green dots that are the ones doing the diffusing, moving for free, then the green dots will move from where there's a higher concentration of green dots on the right, they will move to the left, correct? Where there's a lower concentration of green dots. You know why? Because the green dots will be going down their concentration gradient from right to left. But in the case of water, when water is moving, which way is water going to move? Well, here's what you need to understand. Are you ready? Water is going to want to diffuse as well. But do you remember what diffusion means? Diffusion means a substance will go from where that substance is high concentration toward where that substance is low in concentration. Does that make sense? So what you need to understand is that if the water is the thing moving, the water is going to go from where water is high concentration to where water is low concentration. Did you follow me? Water will go from where water is high to where water is low. sugar will go from where sugar is high concentration to where sugar is low. So look, sugar was high on the right, high concentration on the right, so sugar wanted to diffuse to the left. Water, on the other hand, which side's water higher concentration on? Is water concentration higher on the left, or is water concentration higher on the right? Think about it. Water concentration's higher on the left. You know why? Because there's less sugar. The more sugar you have, the less water you have. Do you see what I'm saying? Like if I have super sugary water, how pure is that water? You see on the right, I have very sugary water. That means I have a low water concentration. That means my water concentration on the left is actually quite a bit higher. You see what I'm saying? In fact, if I had no sugar at all, then I would have 100% pure water, right? Concentrated water, you see? So water is going to go from where water is high and diffuse to where water is low, whereas the sugar will go from where the sugar is high to where the sugar is low. And if you want a general rule of thumb, this will help you. A general rule of thumb, whichever way the sugar wants to go, the water always wants to go the opposite way, right? And it doesn't have to be sugar. If the dye molecules want to go right, the water will want to go left. If the salt wants to go to the right, the water will want to go to the left. If the sugar wants to go to the right, the water will want to go to the left. So whichever way, in general, whichever way the solute wants to diffuse, because the solute's going from high concentration solute to low concentration solute, the water will want to go the other way because the water will be higher concentration on the other side and it'll want to go in the opposite direction. So hopefully that helps you. So by the way, this is called an osmosis problem. Osmosis is the diffusion of water across a membrane. You see, osmosis is the diffusion of water across a membrane, or solvent. Osmosis is the diffusion of solvent across a membrane, whereas, remember this one, dialysis is the diffusion of solute across a membrane. Okay, solute across a membrane, dialysis, solvent, namely water across the membrane, osmosis. And when do you have osmosis problems? Usually whenever you don't allow the solute to cross. If you allow the solute to cross, it's usually a dialysis problem. If you don't allow the solute to cross, now it becomes an osmosis problem. Now only the water can move. Now you have to ask yourself, where's the water concentration high? Because the water is going to go from where water is high concentration to where water is low concentration. Does that make sense? Okay, so this leads me to my last concept of this video, which is Chapter 5, Part 1. And that is this concept of tonicity. Tonicity, the ability of surrounding solution, basically concentration of solution, to cause a cell to gain or lose water. So let me show you some examples. Here's some animal cells. Okay, take a look here, animal cells. Now, let's say I took blood cells. These actually are blood cells from an animal. Let's say I took blood cells and I put them in different solutions. Here's solution 1, here's solution 2, and here's blood cells in solution 3. The thing that's different between these solutions, hypertonic solution, isotonic solution, hypotonic solution, the thing that's different is that in each case I'm putting the blood into solutions in an environment of different water concentration. Did you guys follow that? So the hypertonic example here has some kind of water concentration surrounding the cells. Isotonic has a different water concentration surrounding the cells. And hypotonic has another water concentration surrounding the cells. So what do I mean by all that? Let's say this is salt concentration, right? So I have different salty water and I add it to each one of these. Isotonic means, iso means equal. Isotonic solutions or environments are those where the water concentration outside of the cells equals the water concentration inside of the cells. Did you guys follow that? So the water concentration outside matches the water concentration inside. Let me tell you guys something real quick. Your body is not pure water, right? And your blood is not pure water. And if you were to take one of your cells and crack open a cell, that cytoplasm inside is not pure water. There's salts in there and sugars in there and such. Okay, so let's say we're talking about salts, right? Inside of your blood cells, you have a certain concentration of salt. And that concentration of salt is 0.9% salt. Okay, by salt I mean sodium chloride. Okay, so if you have 0.9% salt inside of your red blood cells, how much salt is outside of your red blood cells in an iso or equal tonic environment? How much salt concentration is outside? Okay, if you have 0.9% salt inside and you'd have to have 0.9% salt outside. Does that make sense? Now let me find my board. Let me grab my whiteboard so I can explain this on a whiteboard. It might help you to visualize it a little bit better, and I will get back to that just a second after a quick break time. Let me take a quick little break time, grab my whiteboard for you. All right, welcome back. So I got here a whiteboard set up to explain iso, hypo, and hypertonic solutions and their effects on cells. Not just any cells, animal cells versus plant cells. There's actually some things to consider here. So let's talk about what I was saying earlier. What happens to a red blood cell in isotonic solution? Remember, isotonic solutions. In this case, you have a red blood cell. Hopefully you can see all that. A red blood cell, and red blood cells have what concentration of salt inside? I said 0.9% salt. NaCl. Okay. So for a solution, a surrounding solution, the solution around the cell to be iso, equal tonic, equal tonicity to what's going on inside of the cell, what kind of salt concentration needs to be outside of that? The same, 0.9% NaCl. NaCl. So do you know what that means? These tonicity problems, what you need to understand about these tonicity problems is that they are essentially osmosis problems. Do you remember osmosis? Osmosis means the solvent, the water is crossing the membrane. The solute does not cross the membrane in tonicity problems or in osmosis problems. Does that make sense? So what's the solute? The solute is sodium chloride, salt. Is the salt crossing the membrane here? In this example, is the salt crossing the membrane? No, the salt is not crossing the membrane. You know what is crossing the membrane? The water. Now let's just say for instance, just for an example, if the salt could cross the membrane, which way would the salt cross? Well, there's a 0.9% salt outside, there's 0.9% salt inside, so salt, there would be no net flow of salt. And that doesn't matter anyway because only the water's crossing, so the water concentration is also equal outside of the cell and inside of the cell. So you know what that means? Let me show you real quick. Let me use the black color marker here. That means water, H2O, for every 1 water that goes in, how many water come out? 1. So what you need to know is that in isotonic conditions, there's an equal concentration of water inside of the cell as outside, so there's no diffusion that really takes place. There's no net flow of water because you've already reached equilibrium. You're already at equilibrium. Does that make sense? By the way, this is why it's important. If you ever go, just this is one reason why it's important that if you ever go to the doctor and you need an IV, to rehydrate your body fluids, that IV contains saline, which means 0.9% salt. That IV isn't pure water or super salty water. The IV that the doctor gives you or the nurse gives you, that IV is actually salt water, and not just any concentration of salt, an isotonic concentration of salt, because you don't want your blood cells to lose water or gain water, right? Does that make sense? So speaking of that, what happens if I take my red blood cell and let's say I've got my red blood cell, and again, how much water concentration is inside? Well, how much salt concentration? Same thing, 0.9% salt inside. Okay, but let's say in this case it's in a hypertonic environment. What does that mean? That means that the water concentration is much less outside. So what does that mean? So for example, what if I have 1.8% salt outside in the surrounding solution? 1.8% salt. Okay. Now again, if the salt could cross, which way would the salt go? Into the cell or out of the cell? Into the cell, right? Because the salt, if the salt could cross the membrane, the salt would go from outside where it's higher concentration to inside where it's lower concentration down its concentration gradient, right? But remember, are these tonicity problems? Dialysis problems or diffusion problems? They're diffusion. I'm sorry, they're osmosis. Are they dialysis or osmosis problems? They're osmosis problems. Osmosis problems. So what can cross the cell? Can the salt cross the cell membrane? No, water crosses. So now you have to ask yourself, where is the water high concentration? Not where is the salt concentration high. Where's the water concentration high? Here's a hint. Wherever the salt concentration is higher, the water concentration is lower. So knowing that, where's the water concentration higher? That's right, the water concentration is higher inside of the cells. Inside of the cell. So water will move outside of the cell. Does that make sense? In this case, there's a net flow of water outside of the cell. You know what that means? That means more water will leave the cell than enter the cell. And you know what that causes the cell to do? The cell will shrivel up like a raisin. Okay, the cell will shrivel. The cell will shrivel up. And this is called a crenated cell. Let me write that in another color. Crenated. There you go. Crenated means shriveled up cell. Why did the cell shrivel up? Because a bunch of water left the cell. Why did a bunch of water leave the cell? Because there was a higher water concentration inside of the cell where there's only 0.9% salt than outside of the cell where there's a whopping 1.8% salt. So the water left the cell. Okay, there was a net efflux, a net outflow of water, and that resulted in a shriveled up cell, a dehydrated cell. That's called a crenated cell if we're talking about animals. Now, in an isotonic, this is just a normal cell. This would be called a normal cell. Remember, the isotonic is a normal cell. The hypertonic results in a crenated or shriveled up cell. Why? Because a hypertonic environment or a hypertonic condition means like for example, a very salty environment, putting your cells inside of a very salty environment or very sugary environment. That's what hypertonic environments are. So what about hypo? Okay, what does hypo mean? Okay, let's say I took here's an example of a hypotonic environment for an animal cell. Let's say I took my red blood cell. I take my red blood cell, which has how much salt inside? 0.9% salt, NaCl. And let's say I put it in pure water, right? 100% pure distilled water. You know what that means? That's 0.0% NaCl. 0%. 0% salt outside, 0.9% salt inside. Which way is the water going to want to flow? Obviously, water would be more pure, more concentrated outside of the cell. Water would net flow in. You see with the arrow? Water would net flow into the cell. And this would cause the cell to gain in size, gain mass until it pops. You see? I'm going to write pop. Right? So the cell will actually pop. That's called lysed. So the cell will pop open, and that's called a lysed cell. Lyse means break. The cell will literally break. Lysed. Okay? A lysed cell means a popped open or broken cell. Why did it pop open? Well, because there was a higher water concentration outside of the cell, a lower water concentration inside of the cell. Water net flowed into the cell, causing the cell to grow and grow and grow until it popped. Why did it pop? Well, because we don't have a cell wall, do we? Our cells don't have a tough cell wall. And so it's like blowing up a balloon too much. What happens when you blow up a balloon too much? It will pop at some point. You see what I'm saying? So that's how. You see what I just covered? Let me show you. Look at the slides here. You see this image here? This is the image. This is the image I'm talking about. So look, your blood cells under isotonic conditions, that means the water is equal. Water concentration outside equals the water concentration inside, so there's no net flow of water. For every one water that enters, one water exits. This is called a normal blood cell. The next one I showed you was a hypertonic environment. This means that there was a lower water concentration outside. So for example, remember the 1.8% salt concentration outside? That's a high salt concentration outside. You only had 0.9% salt inside. So, but again, the salt's not going to enter the cells. Instead, the water leaves the cells, causing the cells to shrivel. And then you had the hypotonic environment where, in this case, the water concentration outside of the cells is actually higher than the water concentration inside of the cells. An example of that would be like, for example, pure water. Water would net diffuse into the cell through osmosis, causing the cell to swell, and then, there's a net inflow of water, which causes it to lyse. Okay, so shriveled up cells are called crenated cells. Normal cells are just normal because they're not gaining or losing mass due to water inflow or outflow. And these cells are called lyse cells. Lyse cells are cells that have ruptured due to too much water inflow. Okay, now do you want to know a trick to remembering this for the exam? Okay, you want to know a trick? Hypo. Here's the trick I used to use. Hypo sounds like hippo, right? Hippopotamus. Hypo sounds like hippo. Hippos are quite swollen, and they do kind of look like they're about to rupture, right? So hypo sounds like hippo. Why would that help me on an exam? Well, if you remember that hypotonic conditions, hypo sounds like hippo. Hippo swells, so these cells swell up. Why would cells swell up? Well, that must mean water's coming in. See, I'm using deductive reasoning. Water must be coming in. Why would water come in? Well, water must be higher concentration outside of the cell than inside. So water comes in, that causes the cell to swell and explode. So just by remembering that hypo sounds like hippo, I was able to deduce the rest of the facts. That, okay, hypo sounds like hippo. Hippos are swollen. What would cause cells to swell? water is flowing in. Why would water flow in? Because water is higher concentration outside than inside, so it diffused inwards, right, through osmosis. Okay, so hopefully that helps you on the exam. One last thing to touch on for this lecture, part 1 of 2 for chapter 5, is what happens to plant cells under the same conditions, under isotonic, hypotonic, and hypertonic conditions. Okay, so what happens to plant cells? Okay, well, how do plant cells fare in this case? Let's talk about that. So a plant cell, obviously, you have. Let me show you something. In a plant cell, you have a plasma membrane. Okay. And you also have a cell wall, a much thicker cell wall. You see what I'm saying? Let me draw this for you, show you what I'm talking about. So, and I'll even draw the little chloroplasts inside, like 4 of them. Look. See, that's a plant cell. Hopefully you can see the blue line. The blue line represents the plasma membrane of the plant cell. The thick green stuff would be the cell wall, the cellulose cell wall of the plant cell wall. And the green dots are the chloroplasts inside, right? The chloroplasts. So a plant cell, believe it or not, a plant cell, an isotonic condition, let's say the plant cell has 0.9% salt, 0.9% salt inside. An isotonic would mean how much salt is there outside? That's right, 0.9% salt outside. So what that means is that there is the same concentration of water inside and outside. So for every 1 water that enters the cell, 1 water leaves the cell. But believe it or not, even though animal cells prefer this, plant cells actually don't prefer this. Plant cells, this cell is called a flaccid cell. Okay, this is called a flaccid cell. And flaccid means droopy. Okay, and plant cells actually don't prefer isotonic solutions because that causes the cells to kind of get droopy. Okay, instead, do you know what plant cells prefer? They prefer hypotonic solutions. Hypotonic. Let's talk about that. So in this case, what's happening? You've got your plasma membrane. You've got your chloroplasts inside. Okay, and you've got your really tough old cell wall. Okay. You got your cell wall here. It's really tough. Okay, look. Hypotonic. Do you remember what hypo means? Hypo is like hippo. So even though, for example, you have 0.9% salt inside, 0.9% salt, NaCl inside. In a hypotonic condition, how much salt would be outside? Well, less than 0.9% salt, right? So let's just say 0 salt. So outside would be 0.0 NaCl. So in this case, which way is the water flowing? Water would flow into the cell. In hypotonic conditions, water flows into the cell, and the cell gets firmer and firmer, and it becomes what's called turgid, okay? Turgid. Turgid is a firm cell, a high pressure internal. There's high pressure internally, high pressure cell. But the cell doesn't lyse like an animal cell lyses, right? Animal cells lyse when it's hypo. Plant cells become turgid, and they actually love it. This is the preferred state for a plant cell. Why don't plant cells explode? How come the plant cell doesn't lyse? How come the plant cell just becomes turgid and it actually enjoys itself, whereas our poor blood cells exploded? Well, remember that cell wall we talked about? That cellulose cell wall? That stuff's tough. Super tough. Okay? The pressure's not going to get to this cell because the cell wall will keep it from exploding. Animal cells don't have a cell wall, so it's like a balloon, right? Balloons pop, whereas plant cells are more like a tire on your truck, right? The tire is too tough to just blow up with just you blowing into it, whereas in a balloon will easily pop with you blowing into it. Okay, kind of that's a good analogy. So what about a hypertonic, right? Hypertonic environment, what happens there? Let me tell you. So, we're almost done with this video, guys. We could do it. Let me show you what I'm talking about. All right, look at this. So, let me show you kind of what's happening here in this plant cell. So, hypertonic, do you remember what hyper means? You still have 0.9% salt inside. You still have 0.9% NaCl, 0.9, that doesn't change, percent NaCl inside. But what would a hypertonic environment do? What kind of water concentrations outside? And what you need to know is that there's less water concentration outside than inside, which means there's higher salt concentration outside. Does that make sense? So outside would be, for example, 1.8% salt. That's more salt, right? More salt outside, less salt inside. But the salt's not crossing, otherwise the salt would come into the cell. What crosses? The water. The water will go from where it's more pure inside of the cell to where it's less pure outside of the cell. So the water is going to leave the cell. Does that make sense? It's going to net flow out of the cell. But does that mean that the whole cell is going to shrivel, including this tough cell wall? Is the tough cell wall going to shrivel up? No. You see what happens? Let me show you something special. You remember the blue plasma membrane? The blue plasma membrane? Only that shrivels. You see that? Look. You see what I drew there? The blue? The blue is the plasma membrane. You see the blue was nicely pressed against the cell wall. You see that? The blue is the plasma membrane is nicely pressed against the cell wall. Look what happened in the hypertonic environment. Yeah, because water leaves the cell and the cell dehydrates. But the cell wall is too tough to shrivel up, but the cell membrane will shrivel up. Do you see that? So what happens is this is called a plasmolized. Let me write that down here. Plasmolized. A plasmolized cell. Plasmolized cells are basically plant cells in salty environments or sugary environments where water's leaving the cell. The cell wall doesn't shrivel up. It can shrink a little bit, but it doesn't shrivel up. But what does shrivel up? The plasma membrane inside, and that takes the chloroplasts with it, by the way. The chloroplasts kind of bunch up as well because the chloroplasts are inside of that plasma membrane as well. Remember here? Look at the chloroplasts. They're all spread out nicely, the green dots. But here, the chloroplasts are all kind of gonna get bunched up. You see? The chloroplasts are gonna get bunched up together because the plasma membrane is gonna smush them together, right? Imagine if you have a box, like a cardboard box, right? You have a cardboard box. That's the cell wall. And then I have a balloon inside. I blow up a balloon inside of a cardboard box. That's a plant cell. The cardboard box is the cell wall. The balloon I'm blowing up inside of the box is the plasma membrane. If I let air out, that's like letting water out. If I let air out of the balloon, does the whole cardboard box shrivel up? No, just the membrane, just the balloon inside of the box shrivels up. So that's what we're talking about here. Take a quick look. Look, isotonic environments, plant cells, that means the water concentration outside equals the water concentration inside. For every one water molecule that enters, one water molecule leaves. So there's no net flow of water. This is a delta G equals 0 situation. And actually, plant cells don't like this. Plant cells, this is called a flaccid cell. This is when your plant cell becomes droopy, right? The plant's not getting enough water, it droops because those cells aren't being as firm as they want to be. Remember, plants don't have a skeleton like you and I have skeletons. Plants don't have skeletons. So plants, they want to grow up strong and tall and rigid, right? And have some support to them. How do they do that without a skeleton like you and I have? Well, the way they pull that off is each and every one of their cells is firm and strong, right? So they can make up for not having a skeleton, right? So when you don't water your plants enough, the plant's not getting enough water, it actually becomes kind of droopy or flaccid. It doesn't like that. It prefers this, right? Look on the right. This is called hypotonic. Remember hypo, hippo? Water's rushing into the cells. And why is water rushing into the cells? Because the water concentration in a hypotonic environment, the water concentration is higher outside, the water concentration is lower inside, so there's a net flow of water in. This builds up a lot of pressure inside of the cell. This is called the turgid cell. Turgidity is the pressure inside of the cell. High turgidity cell has, a lot of pressure inside. And do you remember that central vacuole? It fills up with fluid as well, helping to build more pressure inside of the cells. And plants love this. They dig it up. And the reason they don't rupture, they don't blow up and lyse like our cells, is again because of this really super tough cell wall. And do you remember that not only do plant cells have a cell wall, which in this case is drawn black here, the cell wall, but we also have a plasma membrane. You see this thin green line they drew under the black cell wall? They drew a thin green line. That thin green line represents the plasma membrane of the cell, and that plasma membrane is pushed really firm right up against the cell wall. Okay, now what happens when I put my plant cell in a hypertonic environment? Look at this at the top left, hypertonic environment. This is for example, a very salty situation, very salty environment. What happens to my plant cell? Look, there's a lot of water leaving the cell. Why? Because hypertonic means, the water concentration outside of the cell is very low compared to the water concentration inside of the cell. So the water is going to diffuse out. There's a net diffusion of water out of the cell through osmosis. This is going to cause plasmolysis. Remember what plasmolyzed cells do? The cell wall is not shriveling up. Remember my cardboard box and balloon example? If I let air out of the balloon, only the balloon shrivels up. So in this case, the balloon is an analogy for the cell membrane. You see this green line here? You see this? This is the cell membrane, the plasma membrane of the cell shriveling up. You see this? Look. The plasma membrane and all the cytoplasm and the central vacuole and all the chloroplasts, they're all kind of bunching up together because the cell membrane is all getting dehydrated and the cell's getting dehydrated. And basically the cell membrane's peeling. Look. Look at here. Look here. The cell membrane is taut. It's right up against the cell wall here. Look, the cell membrane is right against the cell wall. It's like pushing firmly against the cell wall in the hypotonic condition. But look here. Yeah, that's right. The cell membrane has peeled away from the cell wall. The cell membrane has peeled away from the cell wall in this case. The cell wall is still there because it's too strong to shrivel up. And this is called a plasmolyzed cell. Okay, so that's a good place to stop with these forms of tonicity, the effects of hypotonic, hypertonic, and isotonic environments. Remember, these are osmosis problems and not dialysis problems. water is going to diffuse from where water is high concentration to where water is low concentration. And that's what these problems are all about. How does the water concentration affect a cell? And that cell gaining water, losing water, or staying the same. All right, I'm just about out of voice, but that's about all I got for you for Chapter 5, Part 1 of 2. Expect the next part soon. Thank you for watching and tune in next time.